Almost every manifestation of the disease can be traced back to dysfunction of a single protein known as the Cystic Fibrosis Transmembrane Conductance Regulator, or CFTR. CFTR is an integral membrane protein composed of approximately 1,480 amino acids and functions as an epithelial anion channel. Its principal role is to provide a passive pathway for the movement of chloride and bicarbonate ions across the apical plasma membrane of epithelial cells. The direction of ion movement depends entirely on the prevailing electrochemical gradient. Therefore, unlike ion pumps, CFTR does not actively transport ions against their concentration gradient. Instead, it behaves as a gated channel whose permeability determines the movement of chloride and bicarbonate between epithelial cells and the luminal surface. An important concept is that although chloride movement through CFTR is passive, the channel itself requires energy to function. CFTR continuously alternates between open and closed conformations through a process known as channel gating. This gating mechanism is regulated by the binding and hydrolysis of adenosine triphosphate, or ATP. ATP is therefore not used to pump chloride across the membrane. Rather, ATP provides the energy required for conformational changes that open and close the channel, allowing passive ion movement according to electrochemical forces. CFTR is predominantly expressed on the apical plasma membrane of acinar cells and numerous secretory epithelial tissues throughout the body. Its physiological function extends far beyond chloride transport alone. By secreting chloride and bicarbonate into the lumen, CFTR establishes an osmotic gradient that allows water to follow passively through adjacent pathways. The result is adequate hydration of exocrine secretions. This hydration determines not only the volume of glandular secretions, but also their electrolyte composition, viscosity, flow characteristics, and ultimately their ability to be transported effectively through ducts and across epithelial surfaces. The respiratory tract provides one of the best examples of CFTR physiology. Covering the airway epithelium is a thin layer of fluid known as the periciliary fluid layer, or PCL. This fluid surrounds the cilia and provides sufficient depth for their normal extension and coordinated beating. Effective ciliary movement continuously propels mucus towards the upper airway, a process known as mucociliary clearance, which serves as one of the most important innate defense mechanisms protecting the lungs from inhaled microorganisms and particulate matter. Normal CFTR activity is essential for maintaining the depth of this periciliary fluid layer. Continuous secretion of chloride and bicarbonate into the airway lumen draws water onto the epithelial surface, preserving adequate hydration of the PCL. When CFTR function is lost, chloride and bicarbonate secretion decrease, water movement correspondingly falls, and the periciliary layer becomes progressively depleted. As the fluid layer thins, the cilia lose the space necessary for effective movement and gradually collapse against the epithelial surface, rendering mucociliary clearance ineffective. With the cilia effectively plastered against the airway surface, thick mucus can no longer be transported efficiently toward the upper airway. Instead, mucus accumulates within the bronchial tree, progressively obstructing airways and creating an ideal environment for persistent bacterial colonization. CFTR also plays an important role within the submucosal glands of the respiratory tract. It is expressed predominantly within the serous acinar cells, where it contributes both to the formation of glandular secretions and their extrusion onto the airway surface. In cystic fibrosis, mucus becomes trapped within these glands because adequate hydration cannot be achieved. Instead of being expelled normally, increasingly viscous secretions accumulate within the glandular ducts before eventually emerging as dense mucus plugs firmly adherent to the airway epithelium. These mucus plugs become heavily infiltrated by inflammatory cells and bacteria, producing mucopurulent secretions that cover and essentially bury the airway surface. Over time, infected mucus remains tightly adherent to the epithelium, preventing effective clearance and maintaining continuous exposure of the airway wall to inflammatory mediators and bacterial toxins. Progressive airway obstruction and chronic infection eventually disrupt normal pulmonary architecture, producing irreversible structural damage that characterizes advanced cystic fibrosis lung disease. Exactly the same pathogenic principle applies to other exocrine organs throughout the body. Within the pancreas, bile ducts, intestinal glands, and other secretory tissues, chloride and bicarbonate secretion through CFTR normally establish the driving force for fluid and electrolyte movement into the glandular lumen. Adequate hydration gives mucins and other exocrine products their appropriate rheological properties, allowing secretions to remain fluid enough for transport through narrow ducts and channels. Another defining feature of cystic fibrosis is an intense, persistent neutrophilic inflammatory response. Large numbers of neutrophils infiltrate the airway lumen and release proteolytic enzymes together with reactive oxygen species and other oxidants. Although these molecules are intended to eliminate bacteria, they simultaneously injure surrounding pulmonary tissue, leading to progressive airway remodeling and ultimately irreversible bronchiectasis. Resident macrophages and other immune cells further amplify inflammation by producing numerous pro-inflammatory cytokines, sustaining recruitment of neutrophils, and perpetuating a vicious cycle of mucus retention, infection, inflammation, tissue injury, and progressive loss of pulmonary function. Abnormalities in airway surface fluid composition also contribute to impaired host defense. Because CFTR normally secretes bicarbonate, defective CFTR activity results in reduced bicarbonate concentration and a lower airway surface pH. This acidic environment diminishes the efficiency of endogenous antimicrobial mechanisms, impairing bacterial killing and facilitating persistent colonization. Moving from pathophysiology to molecular genetics, more than 2,000 different CFTR allelic variants have now been identified worldwide. However, only several hundred of these mutations have been conclusively established as disease-causing. Distinguishing pathogenic mutations from harmless polymorphisms can sometimes be challenging, particularly when novel sequence variants are identified. To address this problem, specialized databases such as the CFTR2 resource have been developed to catalog mutations with clearly established pathogenic significance and facilitate genotype interpretation. 6 major classes of CFTR mutations have been recognized, each affecting a different stage of protein synthesis, processing, trafficking, channel function, or membrane stability. Class 1 mutations result in absent CFTR protein synthesis. These mutations are typically nonsense mutations or large gene disruptions that introduce a premature termination codon, preventing production of a full-length functional protein. Common examples include G542X, R553X, and W1282X. The W1282X mutation is particularly prevalent among individuals of Ashkenazi Jewish ancestry and represents one of the predominant cystic fibrosis genotypes encountered in Israel. Class 2 mutations produce a protein that retains some intrinsic channel function but folds incorrectly during synthesis. Cellular quality control mechanisms recognize the abnormal protein within the endoplasmic reticulum and prevent its transport to the plasma membrane. Instead, the protein undergoes endoplasmic reticulum-associated degradation through the proteasome. The classic example is the F508del mutation, representing deletion of phenylalanine at amino acid position 508. This single mutation accounts for approximately 85% of defective CFTR alleles in the United States and is by far the most common disease-causing variant. Class 3 mutations differ fundamentally because the protein successfully reaches the apical membrane but fails to function appropriately once it arrives.
The defect lies in channel gating rather than protein trafficking. The best known example is G551D, in which glycine is replaced by aspartic acid at position 551. Although the channel is present on the cell surface, it cannot open effectively, resulting in markedly impaired chloride and bicarbonate transport. Approximately 4% of patients with cystic fibrosis possess at least one G551D allele.
The remaining mutation classes involve other functional abnormalities. Class 4 mutations impair conductance through the ion channel pore despite appropriate membrane localization. Class 5 mutations reduce the amount of CFTR protein synthesized, usually because of abnormalities affecting RNA splicing or promoter function, resulting in fewer functional transcripts. Class 6 mutations produce proteins that initially reach the plasma membrane, but undergo accelerated internalization and degradation, reducing the amount of functional CFTR available on the cell surface over time.
And that's our deep dive on pathophysiology and molecular genetics of cystic fibrosis. In the next part, we will discuss the diagnosis and treatment options available for cystic fibrosis. If you found this video valuable, please take a moment to hit the like button and subscribe for more high-yield content. Thanks for being part of our community, and we'll see you in the next video.